up for some questions.
Sure. George Weiner from Iowa. First of all, thank you for an outstanding session. We heard a little bit about use of monkeys and as for toxicology, but not much at all about other large animal models that might be useful moving forward. And pig models, transgenic pig models that are emerging, dog models. A good reason for that is they're expensive and we don't have the reagents. On the other hand, there's a lot of biological questions that we could ask with these models that we can't ask in mice and are too dangerous to do in patients. So I wonder about what your thoughts are about use of some of these emerging models and whether we should invest in development of a pig anti-PD-1 that would allow us to look at novel combinations in pig tumors. I'm from Iowa. I had to ask about pigs now that the candidates are all gone.
I think, you know, there are some limitations in, you know, the historical databases. We have a lot of comfort with the monkeys. We do have a lot of dog studies we get for small molecules, so there are some dog reagents out there. And I think with cardiorenal products, there are actually a lot of pig models. Occasionally, we do get mini pig models from. I think Germany really likes the mini pig, so we'll get non-rodent studies and that kind of species. There's no guidance that said it has to be a monkey. With a lot of the antibodies, it's because you only bind in monkeys, so that, at least for the antibody part, I think that's been a bit of a challenge. If it bound in pigs, and if you could show it was a pharmacologically relevant species, I don't think you would be prevented from using that model. We have gotten rabbits occasionally as well. We don't necessarily like the rabbit plus rat for your general toxicology studies. We'd like something a little more, I guess, further apart on the evolutionary.
I mean, I'm thinking more of efficacy studies with transgenic pigs that develop tumors that can be, you know, imaged in human machines as opposed to just toxicology.
Yeah, well, I get a lot of questions about like companion animal kind of studies as well, but it's been hard for us to leverage those. They're good activity studies, proof of concept. We don't have any problem with that, but we haven't usually used them as toxicology models, partly because they're just not designed as toxicology studies. You know, if you had a rigorous program, I think we would entertain it. We tend to try to be pretty flexible, and I think that CBER is probably even more flexible than we are. But
I mean, I would definitely encourage that field to go forward big time. I mean, they're really doing amazing work with pigs. I think they have skin knockouts now, and they have, which is going to be an amazing tool down the road, considering we mostly have mouse and rat knockouts. We use it in my other life with the other products that I regulate, from, you know, doing skin for wounds and orthopedics and things like that. Models like that could even be used as a potentially as a safety model, depending on, and also cardiac, right? Like Whitney mentions, it can be a really good model. I highly encourage the field to research that area. As far as you know, tumor, cancer, biology, those models just need to be developed and presented to us, and that's part of coming to us early. If you have something like that, come and say, hey, we think this is a really good model to test our system in, and go from there.
yeah, I think you just really need to make your argument and make a clear case. It's not that we don't know. We have to be non-specialists, so you know, you really have to make your justification. We have people from a lot of different backgrounds because we have neurobiologists because we have brain tumors, and we have teratologists to look at our reproductive toxicology studies, but you're not assigned a specific reviewer, so you really just need to make your case.
I wanted to mention that Dr. Alex Snyder actually joined us again on the phone, so if there's any questions that she needs to address, she'd be available to. So why don't you go ahead? Sorry.
Türkan Gardiner from Pragmatica. Although there was mention of the concept of threshold selection for immunization, I'd like to know a little bit more about your experience on how the dose is selected during immunizations. not specifically in treatment. Sometimes in treatment, the concept of highest tolerable dose is used, but do you run into cases like that during immunization as well, especially using animals?
Okay, I'm not sure this one is really directed at me. When we use severely toxic dose in 10% of the animals or highest non-severely toxic dose to look at, to estimate an acceptable starting dose. With the Mabel-based approach, we usually will take some sort of in vitro functional study, maybe T-cell proliferation or IL-2 production or interferon gamma production, whatever is most sensitive for that particular product, and look at an EC20, for example, concentration, and then look at the PK from the animal studies that we have and try to do sort of an estimate. The sponsor often will present this argument, and we'll look at it and agree or disagree and sort of fine tune it. I wouldn't call this really immunization. We don't do, CDER doesn't do vaccines, so I don't know if maybe you were hoping to hear from someone else really on this.
Thank you.
I guess I had the vaccine part, but I think the issue with vaccines and trying to figure out the dose is that they don't appear to be they're not toxic. They don't have side effects. That was a very nice review that Samir had put together looking at, I can't remember how many thousands of vaccine doses that were given. And the essence of trying to figure out things like dose and timing are really problematic. I think the surgery branch went ahead and did vaccinations with up to 1 milligram of GP100 for 52 weeks or 40 weeks to get 40% of T cells recognized in GP100 in one patient. But and I think that it doesn't seem to me to be the major issue in cancer vaccines, at least. I guess the one thing I was kind of interested in coming back is a little more provocative, since just thinking about what limits us from being more aggressive in cancer to push this field in a much more aggressive way that we currently are doing. And I think there is, I think the FDA has been really supportive. I think CBER, a lot of stuff we do is involved with CBER, although some with CDER too. But as you think about the different antibodies that people have developed, and if you think about what happens if you put a different FC in there, or you did something else to that, could you, instead of going back and doing all the other toxicology and development, could you do something where you went to microdosing and did it intratumorally or did things like that? Because the animal models are very important to get basic principles. But the heterogeneity of human cancer, and the more we understand from things like multispectral imaging and be able to assess the dynamic that's going on at the tumor site, to have the chance in patients that have got easily accessible tumors, and with all the other consent issues and things in place, to try and do things like microdosing of combinations or of agents with different properties. Maybe one that would knock out T regulatory cells, or do something else to the myeloid component. I think that's an area where I don't think there's enough money to do all, if you were going to take 10 different variables and look at them in different antibodies to do all the tox, and then the time it would take to do that. And so from the crazy immunology side, I'd like to see some of this move more quickly. And
Hi, John Connolly from Singapore. Just a quick question. Well, first a statement and then a question. One is that one of the things that we've run into with regard to the non-human primate models for testing some of these biologics is a lack of tools for really immunophenotyping. Particularly, it's better than other models and perhaps better than the pig, but it's there's a lot of work that can be done to advance this from the CRO space, and maybe even to come up with academic partnerships to really enhance the capability to deeply immunophenotype some of these impacts. And this will come in really handy when we start looking at combinations of these biologics. And I think this is something that someone needs to really push and work on. The other aspect is really picking up on something that Helen mentioned, that Dr. Fox mentioned, as well as Malcolm. The importance of epitope spreading, and whether or not we can use some of these model systems to assess some of this T-cell therapy, whether it's VST therapy or other kinds of therapy, to try to dip into this neoantigen space that you heard today, and try to optimize concepts of epitope spreading. So coming up with T-cell products that actually would be would have higher adjuvant potential to try to prime some of this epitope spreading responses that you see, and other model systems, maybe even as simple mouse model systems, to optimize this feature of the T cell.
Yeah, I think that's a good point, and we're actually starting to look at whether we get spreading to neoantigens as well, but you've obviously got to do RNA-seq on the tumour and then look for specific neoantigens, but we are starting to do that in the studies.
What about just, adapting your product production pipeline to optimize specifically for this feature of T cell biology, or combinations thereof, such as cellular combinations of DC therapy versus T cell, in order to induce this kind of cross-priming event?
Well, we are getting it with our current products, so yes, we could do some more process development and see if there's any way it could be optimized. But that would, again, the preclinical models for that would be challenging. It would have to be in clinical studies.
And I just actually would ask Helen a question then, because when you were looking at epitope spreading, I think in your data, you weren't looking actually at neoepitopes, or were you? Or those were just overexpressed?
We were looking at overexpressed tumor antigens. We do want to look at neoepitopes, sure, but you would have to know which ones were present in the patient's tumor and then look for them. There are not enough, I think, that are general that you could.
So the concept that we're taking, if you look at the TCGA database for like lung adenocarcinoma, there's 520 patients in there as of last fall. You got gene expression profiles, and you can look at all the genes that are overexpressed in those 520 lung adenocarcinomas. You can go back and say, what are they making responses against? And if you had, if in fact you can use, and I direct you to go to the Journal of Immunotherapy of Cancer, the little review that was put together to talk about biomarker development. But if you could use things like protein arrays against a large part of the proteome to see what you get antibody or IgG antibody responses against, that could direct you towards targets that you have T cell responses against and get you away from looking under the streetlight of just looking at neoepitopes. Not that neoepitopes aren't important, not that we shouldn't be testing them, but to look at what other things we need to look at, because there are a lot of people with cancer who are not going to have mutations that are going to fit into those algorithms to give you epitopes, and we need to find things for them as well.
So I'd like to go back to Bertie's point, and this is more to the FDA colleagues, just to see where the thinking is going, since this is the purpose of such a meeting. So when we look at the immune therapies, vaccines versus immune modulators versus T cells, I'm going to go to the immune modulators first. At the end of the game, the purpose of those is to ramp up the immune system against the tumor. So if anything's going to happen, at least from a side effect point of view, it's going to be immune related. It's going to be a little bit longer term than shorter term than acute. And the question is, since a lot of those cannot be tested in animal models, except if we have specific antibody or large molecule against the animal tissues. And since most of those are long terms, and since we know, at least from preclinical testings, how they work, because that is proof of principle and they tell us how they work, what kind of compartment they activate, all these kind of things. What is the impediment of actually really not having toxicity studies in animal and going into human directly with a safe clinical trial design that would be way more effective than doing the animal studies that at this stage are not that helpful in the first place, right? Did I reflect what you mentioned, Bernie? Yeah.
I think that part of the point of the animal toxicology studies is not just to look for your exaggerated pharmacology, which obviously we're not seeing very well so far with some of the immunomodulators. That's not necessarily true for everything. If you have something like a TLR agonist,
Can you get closer to the microphone?
I mean, that's not necessarily true for everything. If you have something like an innate and a TLR agonist or something, you can see responses in monkeys. They're not useless. I think it's partly a worry that you're going to miss something, that, you know, maybe you don't understand the target as well as, or not you, but like everybody doesn't understand the target as well as we thought we did, and it's expressed somewhere that we just didn't even consider. Especially if you have like an IgG1 with ADCC or CDC activity, that can be an issue. But it's true right now. Most of the time, we're getting studies, and you might have a 500-fold lower dose that you're proposing than from the animal studies because you just don't really see any toxicity, and you really are relying on that in vitro data, a lot of human PBMC data, because we're lucky enough that you can get human PBMCs pretty easily. I don't know if there's a time when we're going to say you don't have to do it unless you have a situation where the antibody really doesn't bind. I think with some of the bispecific T-cell engagers where I don't think that they had a relevant animal model. It was originally done a while ago. They actually did a chimpanzee study, which we would never ask for. And in those situations, we have had times where we didn't ask for a tox model, but if you have something that binds and it's pharmacologically relevant, theoretically, I think it's really hard for us to say, don't do it at this point.
So what would make you say it?
Oh, sorry.
I mean, again, I'm just thinking, what is the thinking process there? Because again, we're just.
And I think it's a worry about off target.
So the issue is, of course, this is a very expensive field. And the more we're doing this, the more expensive it's going to be, and the more difficult it's going to be even developing it. And again, this is one of the concerns of the community, and this is where, I'm sorry, Alan, you wanted to say something?
Yeah, and I think for our products, it's a little bit different, right? I mean, ours is even, we have xeno issues. We really don't have animal models in some cases. We can do a xeno anti-tumor model, but we're not going to see you're not going to be able to see a lot of times a tox study in an NHP. I mean, there's again, I mean, I hate to say this, but it is case by case, it's product by product, and it's science based. And I think you got to come to us and say, hey, this is what's going on. This is what we have. This is the state of the art, and this is what we can do. Oftentimes, for our types of products, which is a CAR T cell, the best you can do is look at off-target toxicities with a bunch of cell lines. And now you have these new cell lines of eye cells coming out, 3D tissue cultures and things like that. We test a bunch of different tissues in an in vitro system. So probably a lot less burdensome, like cost-wise in that case. But that would just, it depends on the product, it depends on the indication, depends on the target. But I definitely think that there are situations where we don't ask for primates and things.
So I'd like to ask Bernie about the last study that you presented about the separation in time. The fact, if I understood correctly, that the anti-PD-1 synergized with the anti-OX40 only if it was given afterward and not concurrently. And if that understanding is correct, obviously adding the dimension of time makes designing clinical trials even much more difficult, as you alluded to, because not only do you have to test every possible combination, but you have to figure out the right order and how far apart they need to be spaced and so forth. So what do you think is the mechanism here? Do you think that anti-OX40 is allowing the infiltration of T cells that then need PD-1 blockade to be able to function, or?
Yes, that's a good question. I'll just start that if you take two different models, one was in the was an MCA205, and the one that I presented where we didn't see, we took the delay to see the effect, is a transplanted PYMT. It's actually the cells isolated from the mammary gland and then gone ahead and transferred into mice and allowed to grow for a period of time. So I don't really know. We've kind of, as good immunologists, we can come up with lots of different ideas into why that is the case. We're currently looking, we've got a paper that's just been accepted in Journal of Immunology, which is applying now a new multispectral approach to look at multiple parameters in mouse tissue and be able to objectively assess numbers. So we're actually applying that to see if there's some differences or if there were differences to start. But I think in general, the MCA205, which is the model they looked at, is one that tends to be less suppressed. It's a fairly immunogenic model. It's been around. It was developed in the surgery branch back in the 90s. So there's a lot of genetic drift in terms of the mouse that it developed in. There's a lot of reasons why it's going to be easier for the immune system to see, and why you'd see more general augmented response would be my impression. But that was work that was done by Amy Moran and Andy Weinberg. Our own data it's complicated. Actually, David Messenheimer will present it at AACR. It got selected for an oral presentation. So we'll have some more data, I hope, to talk about that there. But I don't know. Bottom line is I don't know yet. But I think it is kind of the OX40 effect of boosting both CD4 and CD8 immunity, and then the effect on anti-PD-1. And the data I showed was anti-PD-L1 for them and anti-PD-1 for ours. We've actually done ours with anti-PD-L1 as well, and you see the same advantage to doing the combination with delay with anti-PD-L1.
Are there any other questions? Okay. Well, if not, then why don't we take a break for lunch?